Welcome to Anatomy and Physiology. This is Chapter 5, associated with Week 10 of our term. And with this in mind, this is the last chapter of the term, it's the last lecture of the term, and here we'll be considering the integumentary system. As we look ahead at the material we're going to cover in this lecture, we'll first go through some general definitions associated with skin as we talk through some structures. We'll cover 10 functions of skin, and then we're going to delve into the two layers of the skin, the epidermis and dermis. We'll review a list of tactile receptors found in the skin before we move on to a supporting structure of the skin called the hypodermis, sometimes referred to as the subcutaneous layer. We'll consider skin coloring briefly, then move along to 4 major accessory structures of the skin. And in doing so, we're going to look at hair, sudoriferous glands, which are sweat glands, sebaceous glands are oil glands, before finishing up on the topic of skin with a brief discussion of nails. Our textbook defines the integumentary system as an organ system covering the external surface of the body, while also consisting of things such as our hair, our sudoriferous glands or sweat glands, our sebaceous glands or the oil glands, and our nails, both fingernails and toenails. To appreciate the importance of the skin, let's start our lecture with an understanding of the various functions of the skin, for which I've listed 10 functions here. First, the skin serves to protect, and specifically, we'll see that our epidermis or epidermal barrier protects us from things from the external environment, including biological assaults like bacteria and other microbes, as well as UV light, chemical assaults from toxins or chemicals in the environment, and physical assaults, such as from the bumps, abrasions, and scratches we might experience in our day-to-day lives. The skin also serves as a water barrier, and specifically, the keratinocytes in our epidermis keep fluids in while also keeping fluids out. Ultimately, our epidermis helps create a waterproof barrier of sorts. Another function of our skin is in excretion. Now, while I just said that the epidermis helps create a waterproof barrier, that isn't the full story. The skin, through the help of sweat glands and pores releasing that sweat, provide a site for excretion of certain substances, water, salts, even urea. The skin also serves in absorption. Again, while I just mentioned that the skin provides for a waterproof barrier, we can also rely on skin for the absorption of some things. Think about topical steroid creams, nicotine patches, birth control patches. Aside from pharmaceuticals, we also need to consider toxic materials that can be absorbed by the skin, such as organic solvents, heavy metals, pesticides, and plant toxins like the oils of poison ivy or poison oak. The skin acts as a containment structure, and this simply means our skin acts as a container to hold all the stuff we're made of, including the fluids outside of our cells, in. And while it seems pretty simplistic, it's very critical for survival to be able to maintain and keep these structures and vital substances within the body. Our skin also aids in tactile sensation. Specifically, our skin is highly functional as a sensory organ, as it contains specialized sensory nerve structures that help detect touch, pressure, stretch, vibration, surface temperature, and pain, taking all these sensations and sending them to the brain via the afferent pathway we learned about earlier this quarter, where the brain can then interpret what the body is feeling. We'll look at some of these tactile receptors later on in lecture, as well as we'll return back to them next term in our study of the nervous system. The integumentary system helps regulate body temperature through its tight association with the sympathetic branch of our autonomic nervous system, something we'll look at next term. For now, and based on what we've considered earlier this term, what I want to say is that the skin helps with thermoregulation in 2 ways. First, with the help of sweat glands. When we're warm, we release secretions onto the surface of our skin, which helps cool the body. In addition, when we're warm, we rely on an increase in blood flow to the capillaries found in the dermis and hypodermis of our skin, thus releasing excess heat through the skin to the environment. In contrast, when it's cold, we can reduce the blood flow to these capillaries, minimizing heat loss in order to maintain core body temperature and keep blood closer to those vital organs found in the trunk of our body. We can rely on the integumentary system for the synthesis of vitamin D. Now, vitamin D is both a nutrient we eat as well as a hormone our bodies make. It's a fat-soluble vitamin essential for the maintenance of bone mineralization, and specifically, it helps our bodies retain calcium and phosphorus, both critical for bone building. With this in mind, vitamin D is formed via a series of biochemical reactions that begin in our skin, with a chemical we call 7-dehydrocholesterol, and in the presence of UV light, as well as with the help of our liver and our kidneys, we ultimately go on to produce vitamin D. Our ninth function of the integumentary system is that of having an immune function. Our skin serves an immune function, acting as a first line of defense against microbes in the environment. In addition to that physical shield of the integument, we also have immune cells called phagocytes that are found in the skin and are responsible for engulfing things that shouldn't be found in the epidermis, and then delivering those things to the immune system for presentation and response. Finally, point 10, the skin, and specifically the dermis, serves as a blood reservoir where at any given time the capillaries of our dermis may hold as much as 10% of our total blood volume. Now that you know the functions of the skin, let's go ahead and take a brief look at the various structures of our skin, giving you a preview of sorts. First, I want to emphasize the skin is composed of two major layers called the epidermis and the dermis. Here in this slide, I've also included the hypodermis, as we'll talk about the hypodermis later on in this lecture, given the support it provides our skin. But I want you to keep note, the hypodermis is not part of the skin itself. Be sure to keep it in mind. When I ask you for the layers of the skin, you should tell me we have two layers, the epidermis and the dermis. Now, in terms of this slide, just briefly, as a brief snapshot, the epidermis is located superficially, so we're going to see the epidermis here in this dark brown color, and it consists of an avascular epithelial tissue layer. Then we're going to see the dermis. The dermis is actually composed of two parts in this somewhat pinkish region through here. The dermis is found deep to the epidermis. It is vascular, so it has a blood supply, and it's composed primarily of dense, irregular connective tissue. Finally, in terms of support, we have a hypodermis found here, or sometimes it's called a subcutaneous layer. It's deep to the dermis. It consists of adipose tissue. It helps serve as a shock absorber and an insulator, as well as consists of areolar tissue, which is made up of collagen and elastic fibers. The hypodermis helps anchor the skin to the underlying structures, typically muscle or bone. In this histology slide taken from the PAL site, we see the epidermis here, which is superficial and this darker purple color. The dermis is going to be in pink. We'll see the two layers of the dermis found here. And then we see the hypodermis found in this region with adipose tissue, areolar tissue. We see sensory receptors here. The epidermis, classified as keratinized stratified squamous epithelium, is composed of four principal cell types. We have keratinocytes, melanocytes, dendritic cells, sometimes called Langerhans cells, a type of phagocyte, and tactile cells, a sensory receptor cell that's sometimes referred to as a Merkel cell. I'd like to go ahead and look at all four types of cells in a little bit more detail. First, we have keratinocytes, the most abundant cell type in the epidermis, making up about 90% of our epidermal cells. They arise from the very deepest part of the epidermis, a layer we call the stratum basal, which serves as stem cells for much of the epidermis. And so we would see this layer here, very deep, that stratum basal layer, which gives rise to those keratinocytes. Keratinocytes, with the help of keratin, go on to help prevent water loss. It also helps reduce the entry of microbes. It helps prevent chemical entry from the external environment. And we end up sloughing keratinocytes off each and every day. As we form keratinocytes from this very deep layer, those keratinocytes end up moving up and more superficially until they're found at the surface, and we slough those off. This image here shows desmosomes, those anchors that hold keratinocytes together, and they're found between the lateral surfaces of epithelial cells, serving to bind the plasma membranes of two cells together. Remember, they're described as very strongly adhesive in nature. The desmosomes help with tissue stability, especially in tissues like the integument that experience mechanical stress. Next, we have melanocytes, making up somewhere around 7 or 8% of the total cells of our epidermis. Melanocytes are spider-shaped cells found in the deepest part of the epidermis within the stratum basal layer, and they function to produce a pigment called melanin, one of the primary pigments of our skin. Melanin's major responsibility is to absorb damaging UV light, preventing DNA from being damaged within the keratinocytes. I'll go ahead and point out our melanocytes here, which we see in this brown color, but let's go ahead and look at the next slide because it'll show a melanocyte in much more detail. So again, we're looking at this stratum basal layer of the epidermis. We would see a basement membrane below that, and following that more deeply, we would see the dermis. Melanocytes produce membrane-bound containers or vesicles called melanocytes, which contain melanin. So as we're looking at this spider-like melanocyte here, we see that these dark structures here, and here are going to contain those melanosomes. And within the melanosomes, we find melanin. Melanin needs to find its way from the melanocyte into the keratinocyte. So these cells here in peach are keratinocytes. Those dark circles are the nuclei of the keratinocytes. To do this, melanosomes are transferred via these long, slender processes we find here, or projections, we might call them projections, that project between individual keratinocytes. And melanosomes are ultimately dumped into the keratinocyte, where then enzymes break down the membrane of the melanosome such that melanin granules are released. These melanin granules ultimately form a protective veil covering the nucleus of a keratinocyte, shielding nuclear DNA from damage by UV light. And so here we would see some melanin beginning to cover the nucleus of the keratinocyte. We see that melanin has been delivered here and it's in progress to shield the nucleus from sunlight exposure. We would have more here and here and here as well. Our next cell type is called the dendritic cells, or we might see them called Langerhans cells. These originate in the bone marrow, they migrate to the epidermis, and they act as phagocytes to help activate the immune system and protect the body from microbes as they attempt to enter or invade the skin. They're one of our first line of defense cells, not programmed to find specific things, but rather just be on the lookout for anything foreign to present to the immune system. The dendritic cell here is found in green and lurks in what we call the living layers of the epidermis, and we'll cover that in a little more detail. But we don't find them in the stratum basal, that deepest layer, that mitotically active layer, but we will find them in this next layer of our epidermis that we call the stratum spinosum. Finally, we have tactile epithelial cells, sometimes referred to as Merkel cells. These cells are the least numerous of the epidermal cells, located in the deepest layer of the epidermis, which is the stratum basal, right near the epidermal-dermal junction, where they help protect and interpret touch sensations. Ultimately, tactile epithelial cells or Merkel cells are called touch receptors. And we find one here where we're going to see these yellow fibers. These are nerve fibers that are going to be associated with connecting to these tactile epithelial cells in order to pick up that touch sensation. The image here gives you a better idea of the tactile cell. So we're going to see those Merkel cells here at the very deepest layer of the epidermis. These Merkel cells are in red, and nerve fibers we see project upward to connect and take whatever touch sensation has been received. Ultimately, this moves along this nerve fiber, transferring to the brain, where the brain is going to interpret it, our control center. We've considered the four major cell types of the epidermis, and I've alluded to the stratum basal layer of the epidermis being the deepest layer of the epidermis. Let's go ahead and consider all 5 layers of the epidermis, as well as talk about terms like thick skin and thin skin. Maybe what we should do first is look at the 5 layers of skin by which we move deep to superficial. So again, we've discussed the stratum basal. Our next deepest is the stratum spinosum. If we move more superficially, we're going to see the stratum granulosum, and then we see the stratum lucidum before we find the most superficial layer of the epidermis called the stratum corneum. Now, when we talk about thick skin and thick skin, there's something special about the stratum lucidum. Not all epidermis contains this layer. Specifically, thick skin, which is found on the soles of the feet, the palms of the hand, include all 5 layers of skin, including the stratum lucidum. However, thin skin found covering the rest of the body besides soles and palms is only composed of 4 of the 5 layers of the epidermis, with no stratum lucidum in the skin. Let's go ahead and we're going to look at all 5 of these layers in a bit more detail next. Found in the very deepest layer of the epidermis, we have what's called the stratum basale. This is the deepest layer. It's composed of a single row of cuboidal or potentially slightly columnar mitotically active stem cells, which are responsible for producing keratinocytes. As a stem cell layer, this is a critical layer of the skin. It's highly regenerative in order to continually produce and ultimately replace keratinocytes as the older ones are pushed upward through the epidermal layers close to the surface and ultimately sloughed off. The journey for a keratinocyte begins at this basal layer, so we're looking here, and as we push cells upward more superficially toward the surface, it can take anywhere between 25 to 45 days for that keratinocyte to make its way up to the most superficial level of the epidermis called the stratum corneum. This image here we take from the Powell site. We're showing, and I've highlighted here in blue, our stratum basal layer of our epithelial tissue. Again, we have a single row of cuboidal, somewhat columnar, mitotically active stem cells, which go on to produce our keratinocytes. Remember, other cells found in this region, we see melanocytes producing melanin, as well as our tactile cells, sometimes called Merkel cells. Moving superficially, we go from our stratum basal layer to our stratum spinosum layer, sometimes called a prickly layer, with somewhat polygonal shaped cells. As compared to the stratum basal layer making up just one single layer or row of cells, the stratum spinosum layer commonly is 8 to 10 rows thick. We often find both melanocytes in this region, or at least the tentacle-like structures of melanocytes in this region, as well as we see dendritic cells in this layer acting as phagocytes. Further, we say this layer, the epidermis, is a living layer. So we had that stratum basal that was living as a mitotically active region of stem cells. Our stratum spinosum is also living. And although not vascularized, this layer is close enough to the blood supply of the dermis such that nutrients and wastes can be exchanged with the capillaries found in the dermis, as can our gases like carbon dioxide and oxygen. This image of stratified squamous epithelial tissue shows the stratum spinosum layer here in blue. It's considerably thicker than our stratum basal that we see more deep, and again, it's made up of living cells. Next, superficially, we have what's called the stratum granulosum layer, sometimes referred to as the granular layer because we often see a grainy appearance to this layer. Found in the middle of the epidermis, this layer only consists of a few rows of flattened cells, typically 3 to 5 layers of flattened keratinocytes, actively beginning the process of apoptosis or cell death. Here I say cells are dying. They aren't dead yet, but they're dying. And why is this? Well, first of all, we're moving further away from blood supply, which means it's more challenging for the cell layers to access nutrients, to discard wastes, to exchange gases. Further, we see some important structural changes beginning to take place within the keratinocytes of this layer. First, we see the development of keratin, a waterproofing material, by these keratinocytes. And in waterproofing these cells, it further discourages the exchange of important metabolic substances. Further, we have something called lamellar granules, which release lipid-rich secretions, filling the spaces between the keratinocytes of the stratum granulosum, further contributing toward a water-repellent sealant to help the body prevent fluid loss, as well as help prevent entry of foreign materials. We ultimately see the stratum granulosum marks the transition between the deeper metabolically active layers and the dead cells of the more superficial strata or layers above the granulosum layer. This Powell slide highlights the stratum granulosum layer here in blue. In contrast to the stratum spinosum we saw earlier, this layer is going to appear thinner given the number of layers of keratinocytes as well as a result of the cells beginning to flatten with the loss of nuclei and the loss of cell organelles. Next, we have the stratum lucidum layer, sometimes referred to as the clear layer, which again is only found in thick skin, that is the palms of our hands, the soles of our feet. When present, it consists of somewhere between 3 to 5 layers or rows of flattened dead keratinocytes. Here in our Powell slide, we see the stratum lucidum highlighted in blue, found superficial to the granulosum, spinosum, and basal layers. Finally, we arrive to the most superficial layer of the epidermis called the stratum corneum, or sometimes referred to as a horny layer. This layer of epidermal cells consists of 25 to 30 rows of flattened, dead keratinocytes and makes up the bulk of the thickness of our epidermis. The interior of the cells making up this layer contain mostly keratin, and recall we also have lipids from those lamellar granules of the stratum granulosum, making this a very effective water repellent barrier. Together with keratin, this layer serves as a barrier against different biological and chemical assaults while also protecting against physical assaults, such as abrasion, given the cells of the stratum corneum can be scraped off or otherwise shed. Here in this PAL slide, we see the stratum corneum located superficially and highlighted in blue. We step away from the epidermis to explore the deeper layer of skin called the dermis. The dermis is a strong, flexible connective tissue that functions by connecting the epidermis to the hypodermis, which we'll talk about in a few minutes, and provides strength and elasticity due to the presence of collagen fibers and elastin fibers. It has few cells, including fibroblasts that contribute to producing elastic fibers and collagen fibers. We see macrophages, a particular type of phagocyte. There are a few adipocytes with fat, and on occasion, mast cells or other types of white blood cells may be present. In addition to this, we find nerves, blood vessels, lymphatic vessels, hair follicles. We find particular smooth muscle, sebaceous glands, sudoriferous glands, as well as nail roots. The dermis is made up of two layers, a thinner, more superficial papillary layer, as well as a thicker, deeper reticular layer, which rests atop the hypodermis. Taken from the PAL site, this is going to show, highlighted in blue, the more superficial papillary layer of the hypodermis, and found deeper, we see our reticular layer of the hypodermis as it compares to the 5 layers of the epidermis found superficially. Let's go ahead and explore the papillary layer of the dermis first. This layer is the superficial portion of the dermis, which makes up 15 to 20% of the total thickness of the dermal layer. It includes areolar tissue that has collagen fibers and elastic fibers and helps provide for elasticity and strength. It also includes blood vessels as well as nervous tissue. Now, if you look closely here at the interface, and so we're looking at our epidermis here, by which the very bottom or deepest part of our epidermis is found here in this darker greenish kind of color, we're now going to look to see that our papillary dermis is going to be uneven. We don't have a flat surface. In fact, we have a pattern of ridges and grooves that fit in complement to the epidermis. The projections of the dermis, so we would see a projection here and another here and another here. These are called dermal papillae, which help connect the dermis to the epidermis. And in this manner, we have something called interdigitation, or a puzzle-like connection between two tissues or the layers of two tissues such that they remain connected to one another despite mechanical stress. Further, we find that our dermal papillae projecting up house things like capillary loops, free nerve endings, and touch receptors that we call Meissner's corpuscles that are responsible for transmitting some of the sensations of fine touch and vibration. The palms of the hands and fingers, as well as the soles of the feet and the skin of our toes, we're going to see skin that is distinctly different from skin found in the rest of the body. We've already indicated that this skin, referred to as thick skin, houses all 5 layers of the epidermis. However, this skin is also the site of structures that we call friction ridges, which refer to specialized ridges found on the skin along the lengths of the finger, along the palm of the hand, along the links of the toes, and the soles of the feet. The ridges develop early in fetal development, around months 3 to 4 in utero, as the epidermis conforms to the contours of the underlying dermal papillae. The ridges are designed to produce friction and aid an individual in the sense of touch, thus allowing the hands and feet to grasp surfaces firmly. Friction ridges, which develop from the dermis and project to the epidermis, also contain numerous sweat glands. We call them eccrine sweat glands, a particular type of sweat gland we'll talk about later on in this lecture, with sweat pore openings located on top of these ridges. So why is this worth bringing up? Well, friction ridge skin has been used in the identification of individuals for a number of centuries, and since the 1800s in the form of fingerprints, which are no more than the prints left behind as a result of sweat secretions on those friction ridges. And given the unique and unchanging arrangement of these ridge details of a person's fingers, fingerprint identification is the most positive form of personal identification. It's worth noting here, fingerprints are grouped into 3 different types of patterns, which we see here in this image. We're going to see loops, whorls, and arches, which vary in size, vary in location on the respective surfaces. Loops tend to be the most common ridge pattern, while arches are the least common. To conclude our discussion on the papillary layer, I've included one last image to show the dermal papillae, which we find here if we're pulling away the epidermis from the dermis. We see those dermal papillae projecting upward into the epidermis, making up peg-like projections, or looking a little bit like an egg crate. We move from the superficial layer of the dermis to the deeper layer called the reticular layer. The reticular layer is made up of about 80%, maybe a bit more, of the thickness of the dermis, and we find most of it represents dense, irregular connective tissue. It's further made up of elastic fibers, which provide for stretch and recoil, as well as collagen fibers, which bind water and lend to skin hydration to provide for resiliency. Recall from early in the term, we looked at this particular tissue here, areolar tissue, to consider both our collagen fibers and our elastic fibers. The deepest part of the reticular layer of the dermis, near the hypodermis, has what's called a cutaneous plexus or multiple cutaneous plexi. So we see that right here. This is a network of blood vessels which help nourish the dermis, feed capillaries found in the upper levels of the papillary dermis, providing for diffusion with hair follicles, deep sebaceous or those oil glands, as well as sudoriferous glands. So we see that cutaneous plexus here with extensions that provide for capillaries that move upward along our dermis to feed structures within the dermis, as well as provide for diffusion of substances between capillaries and our epidermis. The reticular layer of the dermis also consists of pockets of adipocytes, contributing to adipose tissue in this layer, which helps insulate the body and conserve heat, amongst other things. And remember from earlier in the term, this was our classical image from the Pell-site, which shows adipocytes. We also find cleavage lines in the reticular layer of the dermis. Cleavage lines, commonly referred to as tension lines or Langer's lines, result from collagen fibers that are oriented parallel to the skin surface. Although they're invisible externally, they're important for surgeons when planning surgeries, as incisions made parallel with cleavage lines are found to heal more readily than when incisions are made perpendicular to the cleavage lines. So for instance, here at this surgical site, if we run an incision that's parallel to the collagen fibers, this is going to be more likely to heal more quickly and leave less scarring as compared to an incision made in this manner, by which we are going to make an incision perpendicular to our cleavage lines. We further have what are called flexure lines. These are creases that appear on the palms of the hands as well as on the soles of the feet, and in these regions we find the dermis is more tightly secured to deeper structures, oftentimes found near joints. Looking at the hand here, the palm of the hand, we have 3 major or primary palmar creases. Named, first of all, we have this one here called the radial longitudinal crease. We have what's called a proximal transverse crease, and then we have a distal transverse crease. These are the 3 major creases. Several additional minor creases may also be present over the palm, but these are the 3 that are common with all palms. Before we move along to the topic of the hypodermis, I want to discuss the various different tactile receptors of the epidermis and dermis of the skin responsible for picking up sensations such as touch, pressure, vibration, maybe itch or tickle. And we talked about some of these already throughout our lecture, but I wanted to put a slide together to show all 5 in one spot. First, we have what are called Merkel cells. These are saucer-shaped, flattened, free nerve endings that make contact with the stratum-based cell of the epidermis, responsible for detecting touch and pressure. And they're common in the fingertips, our hands, our lips, our external genitalia. These are some of the important locations for fine touch, sometimes referred to as discriminative touch. We have Meissner's corpuscles. Meissner's corpuscles are egg-shaped capsules in the dermal papillae layer of hairless skin that aids in fine touch, in pressure, and slow vibration. They're abundant in our hands, especially our fingertips, as well as our eyelids, the tips of our tongue, our lips, our nipples, the soles of our feet, the clitoris of the female, and the tip of the penis for the male. We have Ruffini endings. This is a new topic. These are elongated capsules found deep in the dermis, which we find here, found predominantly in our hands and on the soles of our feet. They're most sensitive to stretching that occurs as our digits or our limbs are moving, providing finger position when gripping objects. Ruffini endings also detect warmth. Then we have our Pacinian corpuscles. Sometimes these are called lamellated corpuscles. These are large oval capsules that are widely distributed throughout the body in the dermis and subcutaneous layers that determine deep pressure and high frequency vibration. And lastly, we have free nerve endings that lack structural specialization, yet support sensing things like itching and tickling. And although we don't see this in our image and it isn't listed here, we also have something that's called the hair root plexus. These are free nerve endings that are going to be wrapped around hair follicles of our skin and are sensitive to touch, such as the movement on the skin surface that disturbs hairs. Like if you have a bug on you or dust lands on you. As you might expect, they're found in hairy skin, and we'll see them further on in our lecture when we consider hair. We move away from the topic of skin temporarily in order to discuss a supporting structure of the skin called the hypodermis, or the subcutaneous layer. Recall from early on in lecture, the hypodermis isn't part of the skin proper, but rather it's the layer of connective tissue found below the skin. Here we find both areolar tissue as well as adipose tissue. These two images are from our PAL site showing typical areolar tissue, typical adipose tissue, by which our hypodermis has much of. Further in this image at right, we see the hypodermis highlighted in blue, found deep to the skin, so found below the epidermis and dermis. The hypodermal layer of the skin has four major functions. First, it pads and protects the body, serving as a shock absorber, as well as helps reduce the risk of damage to the underlying muscles and bony structure. The hypodermis acts as an energy reservoir, storing fat for future energy needs, and related to fat storage, it also provides insulation for the body. Lastly, the hypodermis anchors the skin to underlying superficial structures such as fascia, organs, muscles, and bone. Our textbook makes mention of three pigments and/or structures that help contribute to skin color. We have melanin, keratin, and hemoglobin. I want to look at each of these three to show how these pigments and/or structures provide for skin pigment. First, we have melanin. Recall, and this is a melanocyte, recall the pigment melanin is produced by melanocytes deep in the epidermis at the stratum basal layer, sending melanosomes, which you see here in yellow, along those tentacle-like processes out to the keratinocytes where they're deposited in the keratinocyte, leading to the release of melanin in order to help protect nuclei of living keratinocytes from UV damage. Skin color is due primarily to the presence of melanin. And in healthy individuals, we all have the same rough number of melanocytes found in that deepest layer of the epidermis, with the difference in sun exposure lending to the amount of melanin produced. The more exposure to sun, the more need for a protective shield, the more melanin will be produced by melanocytes. And yet, what accounts for skin color differences? Both light and dark complected people have melanin. What we find out, it's not only the amount of melanin produced, but also the type of melanin that's produced. People with light complexions produce a form of melanin called pheomelanin, which is yellow to red in color, whereas people with dark complexions primarily produce a different type of melanin called eomelanin. I should note here, pheomelanin also contributes to the pinkish parts of the body, like our lips and our nipples, despite melanin production by melanocytes in other parts of the skin. Now, one last thing you might be wondering, what about freckles? How the sporadic, splattered nature of skin coloring is associated with melanin. In fact, people who are born with clusters of melanocytes in turn produce accumulations of melanin that we call freckles. This particular image here shows density of melanin production. We're producing a lot of melanin here, as we can see in these keratinocytes. And with this, in association with sun exposure, by producing more melanin, we're going to have a darker skin color. In contrast, a melanocyte here with little sun exposure won't be producing quite as many melanosomes or quite as much melanin. Thus, we have a lighter complexion generally. Stepping away from production of pigment from cells in the epidermis, we can also consider dietary factors involved in skin pigmentation. Specifically, we have keratin, or our carotenoids, which are involved in skin color, such as the consumption of foods high in beta carotene may lend to an accumulation of this particular pigment in the most superficial layers of the skin, the stratum corneum, and storage in fatty tissues, the hypodermis. While yellow and orange skin may not be desirable, it's often said to make skin glow. These carotenoids from foods listed here, like leafy green vegetables or carrots, tomatoes, sweet potatoes, cantaloupe, squash, these all are going to contribute not only to skin color to make our skin glow, but in addition, they are anti-aging and contribute towards skin repair, where carotenoids have been shown to stimulate fibroblasts to synthesize collagen and elastin production, improving skin elasticity and firmness. Hemoglobin, an oxygen binding protein associated with red blood cells, is yet another contributor to skin color. While not a pigment directly, in pale skinned individuals with little melanin and a somewhat transparent epidermis, the crimson color of hemoglobin associated with red blood cells to carry oxygen circulates through dermal capillaries and may contribute to a pinkish skin hue. We now move from skin to the accessory structures. Sometimes we call these the epidermal derivatives or the appendages of skin. And we're going to see three major components. We are going to see hair, we'll see exocrine glands, of which we have two types, as well as we will look at nails. Hair is our first topic, skin appendages or accessory structures, and these are elongated, keratinized structures that form within epidermal invaginations, which we call the hair follicle. The color, the size, the shape, the texture of hair, it all varies according to age, to genetic background, and to the region of the body. All skin has at least some minimal hair, except for the palms of our hands, the soles of our feet, the glans penis for men, the clitoris of women, as well as the labia minora. At different parts of our lives, we might end up seeing one of three types of hair on our body. So we say that there are three types of hair found here. First, we have lanugo, which is referred to as fetal hair, and this is hair that a baby is born with, but it falls out shortly after birth. From there, we develop downy hair, sometimes referred to as vellus hair. This is a thin, fine hair that grows on most of the body of the child, as well as we see in adult females. And finally, we have terminal hair. This hair doesn't begin to come into play until puberty, where it's found to respond to certain sex hormones to grow thicker and darker, as well as in places where it never grew before puberty. This is our adult hair. Terminal hair is found in the genital area, the pubic and anal area, the axillary area found in the underarms, as well as for men on the face and on the chest. And the primary functions of hair is to help warn us of insects or particulates on the skin, to help guard against physical trauma, to protect us from heat loss, as well as to help shield the skin from sunlight, as well as skin damage. We can look at the anatomy of hair. First, we have what's called a hair shaft and a hair root. They're two parts of hair. Again, hair shaft and hair root. The hair shaft is the visible, superficial portion of hair that projects from the surface to the skin. When we take a close-up look at the shaft, and so we're looking at it here, this is our shaft as it's moving outward from the skin, but our hair root is going to be found in this region. When we look at a close-up of the shaft, we're going to see it's composed of three parts: an innermost medulla, a more superficial cortex, as well as then an outer cuticle, and we'll look at that in more detail in the next slide. We also have the hair root, which is found deep to the shaft. It penetrates into the dermis and is surrounded by a hair follicle. That's the sheath of skin and connective tissue, while also being connected with a sebaceous gland or an oil gland, and we'll understand why that is coming up. Looking at the hair shaft, the part visibly protruding from the skin is composed of 3 concentric circles or layers, that medulla, cortex, and cuticle. As we look at the innermost medulla, or the innermost medulla found right here, it's the deepest part. It's a central core composed of large cells as well as some air pockets. Next, we have the cortex, and we see the cortex here as well as see the cortex here surrounding the medulla. These are heavily keratinized and they're very densely packed cells that surround the medulla. And finally, we have the cuticle. When we look at the cuticle here, and we would see our cuticle is going to be in this region right here. We're going to see this is a thin layer of very highly keratinized squamous cells arranged somewhat like overlapping shingles of a house covering the cortex. And so we see that maybe a better view of that right here. In addition to the 3 layers of the hair shaft, we can also look at other structures comprising hair. First, we have the hair follicle. This is found within the skin surrounding the hair shaft and is also made up of 3 concentric parts. Located next to the cuticle, we have the internal root sheath produced by the matrix, which creates a layer of epithelial cells between the hair and then an external root sheath. So moving outward, we have that external root sheath, which is going to cover the internal root sheath, and it extends all the way up to the epidermis. It's actually a continuation with the stratum basale and the stratum spinosum. In the image here at the bottom, we have further what's called a glassy membrane. And so we see that glassy membrane right here. This is just an acellular layer that separates the hair follicle from the dermis itself. And then moving outward from there, we have what's called a connective tissue sheath, which surrounds epithelial cells of the hair follicle. So we have 4 components making up the hair follicle by which we surround the 3 parts of the hair shaft. This image is taken from the PAL site and it shows the various parts of our hair follicle. It's going to show our internal root sheath here. We will then see our external root sheath. We would see a glassy membrane here before we see the connective tissue root sheath. We don't actually see a glassy membrane labeled, but we would know that it falls between the external root sheath and our connective tissue root sheath, all of which are going to be circled around in these concentric rings with our hair shaft. In addition to hair follicle structure, we also have, in a very deep position, the hair bulb and the root hair plexus. The bulb is somewhat of an onion-shaped structure found at the base of each hair follicle, and it's going to house something called the papilla of the hair. So the papilla is this little nipple-like indentation at the base of the hair, which contains blood vessels to help nourish the growing hair follicle. We also have what's called a root hair plexus, and I mentioned root hair plexus when I was talking about some of our sensory receptors. The root hair plexus is a bundle of sensory nerve endings which wrap around the hair bulb, so we see it wrapping around the hair bulb like this. Thus, the root hair plexus serves as a sensory receptor for touch. If we have a mosquito, if we have a piece of dust, if we have something that lands on our skin and touches the hair, that movement of the hair is ultimately going to send a message along via this root hair plexus to the brain that indicates with that message that there is something on the skin. We move away from the study of hair just temporarily to discuss exocrine glands. First, let's talk about glands generally. Recall glandular tissue secretes things. In the case of exocrine glands, we secrete things via a duct or other opening onto a surface. And in the case of the integumentary system, we have 2 major exocrine glands to consider. We have sudoriferous glands or sweat glands, as well as we have sebaceous glands or our oil glands. Now, as we study moving forward, we're going to see that there are 2 types of sweat glands or 2 types of sudoriferous glands, the eccrine and apocrine glands, and then we will study our sebaceous or oil glands. Let's first look at our sudoriferous glands, or the sweat glands, for which we have 2 types, eccrine and apocrine. First, we have our eccrine sweat glands. These are the most numerous and most abundant of our sweat glands and are found widely distributed on the body with a focus on the forehead, the palms of the hands, and the soles of the feet. They're simple, coiled, tubular glands where the secretory part here is deep within the dermis, and then we see a duct move upward through the superficial part of the dermis, through the epidermis, and onto the skin surface. Sometimes we see that eccrine sweat glands are labeled American sweat glands. Those two terms can be used synonymously. Sweat produced by eccrine glands is used to cool us. It's moderated by the sympathetic division of our autonomic nervous system. It's composed primarily of water with a small amount of salts. We have some antibodies and other immune-related protein secretions. We see the release of some metabolic wastes here, like urea, that we get from protein breakdown. The pH of the sweat is somewhere around 4 to 6, so it's acidic. And given that low pH and acidic nature, it's often referred to as an acid mantle, making our skin inhospitable to many microbes that might otherwise appreciate living on our skin. In contrast to eccrine sweat glands, we also have what are called apocrine sweat glands. These are coiled tubular glands as well, found deep in the dermis, sometimes even originating in the hypodermis. Their production of a secretion begins at puberty. It's under hormonal control, and the secretions are discharged into hair follicles associated with hairs found in the underarms, around the nipples, and in the pubic and anal regions. With this in mind, these sweat glands are much more limited in nature as compared to our eccrine sweat glands that we just looked at in our last slide. The composition of this sweat is different than that of eccrine glands as well, with a cloudy, sticky secretion made up of fatty substances and proteins. And while itself is odorless, sweat from these glands, when mixed with organic molecules found on the skin, take on the generally unpleasant smell we recognize as body odor as bacteria break down components of this sweat. And finally, there are two modified apocrine glands I would like you to know. First, we have what are called ceremunious glands. These are modified apocrine glands found lining the external auditory meatus. So we're seeing these glands in the ear canal, where the glandular secretions mix with sebum produced by some sebaceous glands within that auditory meatus to form a sticky substance called cerumen or earwax. This wax is meant to help protect the eardrum while protecting the ear from foreign invaders like bugs and other things. We also have mammary glands, another modified apocrine gland that secretes milk, and we'll study this in more detail in chapter 27 when we look at the female reproductive organs during the third term of anatomy and physiology. Now we turn our attention from sweat glands to oil glands, or more formally known as our sebaceous glands. Sebaceous glands are involved in oil secretion called sebum, and they're found along the surfaces of our body except for the palms of our hands and soles of our feet. And with a few exceptions, they're found associated with hair follicles, where they release their substance into a hair follicle, similar to our apocrine glands, rather than directly onto the surface of the skin, as we saw with our eccrine sweat glands. Sebaceous glands are stimulated by hormones, especially those involved with puberty. The sebum released is made up of triglycerides, so some fats and cholesterol, proteins, and inorganic salts. With this in mind, these secretions lubricate the hair shaft to keep it from becoming dry and brittle, while also keeping the skin soft and somewhat pliable. In association with sebaceous glands, we find what's called the erector pili muscle, a smooth muscle, so not a skeletal muscle, but a smooth muscle, involved in aiding the hair in standing upright when frightened, something we call piloerection. The contraction of the smooth muscle, this erector pili muscle, lends to the release of sebum from the sebaceous gland into the hair follicle, ultimately releasing oil onto the skin's surface. One other thing to mention here, I said that there were a few exceptions by which sebaceous glands were not associated with hair follicles. Specifically, this is in locations like the lips, the glans penis, the labia minora, and the tarsal glands of the eyelid. Here, sebaceous glands open directly onto the surface of the skin rather than affiliate with hair follicles. I think this is actually the best image to show you the erector pili muscle right here, by which, in a process we call piloerection, we will see this hair shaft stick straight up. And this image here is going to show you that sebaceous gland by which sebum can be released into the hair follicle with the help of the erector pili muscle. This slide here is taken from the PAL site, and it's assigned with this week's lab checklist. Notice here we have a cross section of the skin where we're going to see the thin epidermis as well as our much thicker dermal layer, we see that the hair follicle is just the invagination of our epidermis here. We would see our shaft in this area right here. Now, what I want you to take a look at is we have sebaceous glands associated with this hair follicle. So we would see release of sebum in association with the erector pili muscle, which we don't actually see here. It hasn't been stained properly in order to see that particular smooth muscle. Although I mentioned some general functions a few slides ago, let's formally lay out the functions of our sebaceous glands. First, sebum helps lubricate hair to keep it from becoming brittle and dry. Further, it helps keep our skin pliable and soft in order to promote elasticity as well as aid in that waterproofing of the skin surface. And lastly, sebum helps prevent microbial growth. That said, although sebum is essential to keep our skin soft and pliable and our hair shiny, sometimes overproduction of sebum during puberty in response to sex hormones like testosterone can result in the release of excess sebum, which can go on to clog pores, and then mixed with other things like dead skin cells, bacteria contained within the pores can feed off of this mixture of stuff, the dead cells and sebum, resulting in bacterial infection, which can lead to the recruitment of white blood cells to fight infection, ultimately contributing to the formation of acne. We turn to our last topic of this chapter to look at the nail. Nails are skin appendages or accessories. They're made up of plates of tightly packed, hard keratinized cells, which form clear, solid coverings over the dorsal surfaces of our distal phalanges. They're translucent in nature, yet appear pinkish because of the blood flowing in the underlying capillaries of the nail bed. Nails tend to serve three functions. First, they help us grasp things. They help us manipulate objects within our environment. They also provide protection against trauma to the ends of our digits, so as we bump our fingertips, we don't end up hurting ourselves. And then third, they allow us a sharp edge to help scratch or itch body surfaces. Let's go ahead and look at the various parts of the nail. First, we have what's called a nail bed. So if we look at this image here, we see our nail bed here. The portion of the nail visible at the distal end of the dorsal surface of the phalange. It's sometimes called a nail body. If we move here, we're going to look at our nail root. If we move this way proximally toward the body, we're going to see our nail root here found underneath some skin, and we'll have a name for that in a moment. So the nail root is the portion of the nail buried in a fold of the skin. And even further, more proximal, we actually see what's called the nail matrix, a thickened part of the nail bed where germinal cells divide by mitosis to produce nail growth. We have nail folds, where skin folds overlap the borders of the nail. Here we actually see lateral nail folds here and here. So when we're looking at our nail folds, we have what's called an epinecium, more commonly called the cuticle. Here's our epinecium right here. Our epinecium would be found in this region right before we get to our nail root. And this is a narrow band of epidermis extending from, as well as adhering to, the proximal border of the nail wall. Further, we have what's called a hyponychium. We don't actually see it labeled, but the hyponychium is found here. It's found beneath the free edge of the nail, and this thickened region of the stratum corneum secures the nail to the fingertip. Found at the proximal base of the nail here, we have what's called the lunula or lunulae. And this is our little crescent moon. It's the area by which the nail is growing, and it appears white because we have vascular tissue underneath which isn't showing through due to the thickened region of the epidermis in this area. With this last slide of week 10, the last week of the term, I want to wish you congratulations on getting through the first term of anatomy and physiology. I wish you luck studying for your final exam. And if you have questions, don't hesitate to reach out to me during office hours. Meanwhile, make it a great day.